In this section, we'll talk about some other applications of biotech in the field of medicine. The next application that we are talking of is gene therapy. This gene therapy basically means if there is a faulty gene, then that gene can be replaced by a correct gene in order to cure or treat that particular disease. The first clinical gene therapy was performed to treat the defect in a gene which synthesizes ADA, that is adenosine deaminase. Let us first understand how this enzyme works and what is the normal process, and then we'll come to the actual process of gene therapy. Bone marrow cells produce lymphocytes, and these lymphocytes get specialized in thymus or lymphoid tissues, and these cells form B and T cells, which actually are the cells responsible for our immune system. For these bone marrow stem cells to get converted into B and T cells, an enzyme called adenosine deaminase, that enzyme is required. And this enzyme is a protein, so there is a gene which undergoes transcription and translation to form this enzyme. The abbreviation which we use for this enzyme is ADA. So in normal situation, the stem cells which are present in the bone marrow, in presence of this enzyme, that is ADA, divide to form B and T cells. B and T cells are responsible for our cell-mediated and humoral immune system. So when we talk of third line of defense, that is immunity, that is controlled by B and T cells, whether it is cell-mediated or it is humoral immune system. Now, in case if this gene is faulty, then this enzyme will not be synthesized, and if this enzyme is not synthesized, B and T cells will not be synthesized. That means the babies will be born without immune system. This condition is known as SCID. Severe combined immunodeficiency. It is called severe combined immunodeficiency. And if a child or a baby is born without immune system, then there are chances of all kinds of diseases because there is no third line to defend the body. This was the first gene therapy which was performed, and it was performed on a 4-year-old girl who was suffering from this condition, and the year when this was performed was 1990. What the scientists did, they isolated the ADA synthesizing gene. This gene was introduced into lymphocytes. The lymphocytes of the patient, that is of this girl, were taken out, and in this ADA gene was introduced using retrovirus. Retrovirus was used as a vector, and these transformed lymphocytes were infused into the bone marrow of the patient or of this girl. So when she was given infusion of these lymphocytes, temporarily this enzyme was synthesized at that location because lymphocytes were given in that site, that is in the bone marrow where this enzyme is required. There is no problem with the cells, and as soon as these lymphocytes produce this enzyme, B and T cells were synthesized. And as B and T cells are synthesized, the immune system gets revived. But the life of these lymphocytes is short. So such patients are required to be given this infusion on a regular interval, short intervals. If this treatment is performed at an early stage, maybe at a stage of zygote or two-celled stage, then this correction can be permanent. After birth, when this kind of treatment is done, then the only option remains is giving these lymphocytes in the form of infusion. And these lymphocytes were transformed by adding the ADA gene. The lymphocytes were taken from the patient's blood only, and this is how this girl was treated. So this was the first clinical gene therapy that was to treat the deficiency of ADA gene. Now other applications include obtaining various kinds of hormones using the same rDNA technology. The hormones which have been synthesized using rDNA technology, one is growth hormone. We know that deficiency of growth hormone causes dwarfism. So if it is detected at an early stage that the child may get this kind of symptom, growth hormone production is less, then artificially growth hormones can be given to that patient or that child. And this growth hormone would be synthesized by the same process. Other substances which are synthesized, one is tissue plasminogen activator, and these activators are known as TPAs. Other substances which are used or synthesized by the same technique are somatostatins, streptokinase, these biochemical substances are used to treat various conditions in human body. For example, tissue plasminogen activator is used as a clot buster. It is used to dissolve the clot. Certain chemicals are used as immunosuppressants during organ transplantation. One more application is to obtain edible vaccines. We take vaccines which are killed or weakened pathogens, and there is a risk that one pathogen or a few pathogens are not that weak or not properly weakened or killed. In that case, chances of having that disease instead of getting immunized against it are there, though the chances are less. Though edible vaccines have been created, edible vaccines, the protein which acts as an antigen has been introduced into plants like banana and tomatoes, and these bananas or tomatoes, when consumed, we get those vaccines. So edible vaccines are there. Second generation vaccines have also been developed. So vaccines obtained by rDNA technology, they are known as second generation vaccine. First generation are the ones which are typically obtained by orthodox method, that is weakened or killed pathogens. By rDNA technology, the vaccines which we obtain, they are called second generation vaccines, and third generation vaccines are the ones which are synthetic vaccines. So these are some applications of biotechnology or rDNA technology in the field of medicine.